Hello everyone. I hope you are doing well. In continuation to our discussion about the protein's secondary structure, in today's class, we are going to discuss about alpha helix. Alpha helix are the most common motifs which are found in proteins with beta plated sheets. Now, when I am saying with beta plated sheets, I am talking about the common findings. Okay, not necessarily whether they are always be with beta plated sheets. So, in today's class, we are going to discuss about the formation of the alpha helix, how they are important, how their stability is important for the entire stability of the proteins, and what can be the disadvantages of having certain amino acids which can cause the disruption in these particular motifs. So, let's start the alpha helix. Okay, so if we define alpha helix, these are the simplest arrangement of the polypeptide chain which could assume with its rigid peptide bonds. You know, the peptide bonds are rigid because of the double bond character. But there are other single bonds which are free to rotate. And that's why we discussed in the Ramachandran plot about the phi and the psi bond. So we are actually referring here the free bonds as the phi and the psi bonds, and the rigid peptide bonds are the double bond character because it happens due to the resonance. Okay. So the alpha helix, it is a spiral structure which is stabilized by intramolecular hydrogen bonds. Remember, intramolecular hydrogen bonds, and these bonds form between the carbonyl oxygens and the amide nitrogens, which are approximately four residues apart. So you can see here this is the helical structure, and here in one residue this is the CO, and other residue it is the NH group, and they are making the hydrogen bonds. And if we calculate how far they are from each other, so they will be somewhere around 4 residues apart. Okay, so amino acid 1 is hydrogen bonded to amino acid 5, so basically it is n plus 4. If n is 1, n plus 4, that is 5. So amino acid 2 is hydrogen bonded to 6, and in the same manner hydrogen bond, it, it is formed between the residue number 3 and 7, in between them. Okay, so these hydrogen bonds, they are parallel to the axis of the helix. So when we, if we take a imaginary axis in the helix, so these bonds, they are parallel to this axis of the helix, and there are about 3.6 residues in one turn of the spiral. Okay, so if we see the one turn, it will have somewhere around 3.6 residues in a turn, and each amino acid in the helix, it rotates it 100 degrees. So when it rotates 100 degrees, and that's why this spiral is formed. So you can see here the one amino acid, it is rotating somewhere around 100 degrees. It's contributing to 100 degrees. Now to complete a full turn of 360 degrees, we need 3.6 amino acids. Okay, so there must be 3.6 residues per turn. So the average alpha helix which has been determined, it is somewhere around 10 amino acids long, but this is just the average. In some proteins it may be just 5, in some proteins it may be more than 40. So the range from individual peptide to or protein to protein varies, but the average length of alpha helix is 10 amino acid long. Now, a pitch is the distance between the turn, and it has been determined somewhere around 5.4 angstrom. So this is the pitch which is between the distance between these turns. And the rise, this is particularly the rise, is the distance between the amino acids. So in between two amino acids, what is the distance is somewhere around 1.5 angstrom, and that is known as rise. So what is rise, what is pitch, okay, what is, you know, the how the degrees which are rotated by each amino acids, and you know what is the average length of the amino acids. Every data here has been experimentally determined. Now the allowed phi angles is minus 57 degrees to minus 60 degrees. So if you can see the Ramachandran plot, this is our alpha helix, and this alpha helix, if you see the phi angle, here is the phi angle that falls somewhere around minus 57 to minus 60 degrees, and the psi angles are minus 45 to minus 50. So this is the psi angle, this is falling here somewhere around minus 45 to minus 50. So you can see here this is 0, this is 90, and this is coming somewhere in between them, so minus 45 to minus 50 degrees. These are the values of the phi and the psi angles which are the allowed rotations here. Okay, now all of the amino acid side chains, they face the outside of the helix, as it is the most energetically stable arrangement. So you can see here these are, they are the side chains and they are facing outside the helix. So you can see this is the helix and these are, they are facing outside, which is the most energetically stable arrangement. Okay, now to figure out if the helix is right-handed or left-handed, it is very easy. How do you do that? You make fist of your right hand with the thumb outstretched and pointing straight up like this. So this is the right hand and you are just outstretching your thumb and pointing straight up. Okay. Now assume helix spiraling up your right thumb in the direction in which the other four fingers are curled. So you can see here the four fingers are curled here and they are, helix is going upward. So if we start doing this, then it will coming like this, this and so. So it is the counterclockwise. So this is your right hand helix. In any helix you can find out whether it is right hand or left hand. So the left hand will demonstrate a left hand helix. So now instead of using your right hand, you use your left hand, your thumb up, and then fingers are curling, and then you can rotate the helix upward and you will find whether it is the left hand helix, and that will rotate in the clockwise direction. Alright, so that's how we determine whether a particular helix is right handed or it is left handed. So the right hand helix is found in all the proteins. You remember, whatever the proteins we have seen, we have studied so far, every protein has right hand helix and no left hand helix has been found here. So an alpha helix is stable only if hydrogen bonds can form properly. If hydrogen bonds cannot form properly, then alpha helix cannot be stabilized. And the side chains, which are the R groups, they do not interfere with each other. So that means it is very simple that if the R groups, they are not sterically, they are not creating any disturbance, then only an alpha helix can be stabilized and hydrogen bonds can form properly. So there are certain favorable amino acids which you will find in and which have been determined experimentally in the alpha helix. So what are these favorable amino acids? They are methionine, alanine, arginine, glutamate, leucine and lysine. Usually these amino acids, they dominate the alpha helix structure. Okay. Now proline, it cannot participate in hydrogen bond, but it can turn up in any of the first four positions in the first turn of alpha helices. Okay, so it can come within the first four alpha helices, and but it cannot participate in the hydrogen bonding. But if it comes elsewhere in the helix, then it can produces a band in the helix or kink in the helix, and therefore proline is called the helix breaker amino acid. Okay, now you understand why why proline is called helix breaker amino acid, because it produces a kink in the alpha helix. And glycine is another, it is again the simplest amino acid which has H or hydrogen as a side chain. It is also unfavorable in the helix. And why? because it has a wide angles of rotation, because its side chain is the smallest. So when forced into a restricted helical conformation, because helical conformation is a forced structure, right? It is a repeat structure. It is a very, the structure which is stabilized and it has a certain dimensions. So it loses a large amount of conformational freedom, because glycine is, it has a tendency to have the conformational freedom because of the side chain. But when it goes in a helical conformation, and helical conformation is a very restricted conformation, then it loses a large amount of conformational freedom, and its entropy or randomness decreases, and that is known as the entropic cost. So basically it pays the entropic cost when it goes inside the helix, and that's why it is unfavorable in the helix. Okay, so helix is also destabilized by charged repulsions and bulky side chains. So, if there are charged repulsions of the same charged amino acids, or there are some bulky side chains which can cause steric hindrance, that can also destabilize the helix. So, these are some of the factors which actually can destabilize the helix, and if you remove these factors, and if you bring the favorable factors, then only alpha helix will be formed. All right, now why alpha helix is a very stable and commonly formed structure? Because if you see, in among all the secondary structures, it is the alpha helix and the beta pleated sheets which are the most common structures which are found in the proteins. So the question is that why alpha helix is very stable. Okay, now almost every peptide bond, it participate in hydrogen bonding. Okay, got it? Every peptide bond, almost, it participates in hydrogen bonding. Then each turn of helix is connected to a nearby turn. All right, so you can see here, when there is a turn, they are connected to a nearby turn using these hydrogen bonds. Now many weak hydrogen bonds together create strong overall stability. So individually, the hydrogen bonds are very weak, as you have seen in the previous class. But when they collectively, they are very strong. Okay, so it is compact and energetically favorable. So once helix is formed, it is very compact and it is energetically favorable. Now, terminal residues do not always have partner residues which are available for complete hydrogen bonding, and therefore the terminal part of the helix, they usually do not participate in the hydrogen bond. Now what is the consequence of this? We will see shortly. It is like a spiral staircase where each step is connected to steps above and below by many small supports. So, individually they are very small, but collectively they are very strong. And that's why alpha helix is very stable, and it is a very commonly formed structure in the proteins. Okay, and because these operate in short term and they are weak individually, but when they are collectively, they are making the helix and they are making this favorable conformation, then it is making very strong structure, and they stabilize the alpha helix. So hydrogen bonds are the contributors, the major contributors of the alpha helix formation. Now, let us understand what is the helix dipole concept. And you remember that I just told you that the terminal parts of the helix, they usually do not participate in hydrogen bond. Now you know that each peptide bond is polar. Okay, you can see here, this is polar peptide bond, and there is a slight positive charge near NH side. So you can see here, this is the NH side, and you can see here, this is the positive charge, and slight negative charge near the CO side, and this is the CO side, and you can see the partial negative charge. So every peptide bond, it behaves like a tiny dipole. So it is behaving like a tiny dipole. Now in alpha helix, these dipoles align in the same direction, so they add together, and an overall helix dipole is created. So you can see here, all are adding together. This is the negative side, this is the positive side, and there is a dipole moment, and they all are adding together, and an overall helix dipole is created here. So positive is near the amino terminus, which is the N terminus, and negative is near the C terminus, which is the C terminus. Got it? Good. Now the dipole charges, they are exposed at the first and the last four residues because they usually do not participate in the hydrogen bonding. So these charges are exposed there. Therefore, the N terminal is positively charged and the C terminal is negatively charged. And why it happens? Because there is an incomplete hydrogen bonding. Okay. So negative amino acids like glutamate or aspartate, they stabilize the helix at the N terminal. Now you know that N terminal is positively charged, and these amino acids, glutamate and aspartate, they are negative amino acids, and therefore negative attracts positive, so they stabilize the helix at the N terminal. And the same thing happens at the C terminal, which is negatively charged, and we need positive amino acids like lysine and arginine to stabilize the helix at the C terminal. And that's how the helix is stabilized by these positive and negatively charged amino acids. Okay. Now there are 5 different kinds of constraints which affect the stability of the helix. First is the electrostatic repulsion or attraction between the successive amino acid residues with the charged R groups. Okay, easy. Second is the bulkiness of the adjacent R groups. So if there are R groups, bulkier R groups which are adjacent, then they will create a steric hindrance, right? And that will destabilize the helix. So that is again a constraint. Then third is the interaction between the R groups at the 3 to 4 residues apart. Now when we say 3 to 4 residues apart, why? Because you know that the 3.6 residues, they form around a turn. So we are talking about 3 to 4 residues apart when alpha helix is twisted. So the amino acids which are 3 or 4 residues apart, they face each other in a 3-dimensional space. Alright, so opposite charge R groups, they can attract each other and stabilize the helix, but if the situation is opposite, if they are the like charges there, then they will repel, and this repulsion can cause the destability of the or instability of the helix. So hydrophobic amino acids, they stabilize the helix through hydrophobic interactions. So you know these hydrophobic amino acids, they do the hydrophobic packing there, and these interactions, they stabilize the helix. They usually found in the interior. Okay. Now other factor is occurrence of the proline and glycine, which we have already discussed. So proline can produce a kink, it is a helix breaker, and glycine because it has a wide conformation, but if it goes inside the helix, which is a very strict conformatory environment, then it it pays the cost. So that's why the if the proline and glycine by mutation, if they occur within the helix, then that helix will not stabilize. And the last is the terminal dipole and the residues interaction. So at the terminal dipole, the N terminal is positively charged, the C terminal is negatively charged, and we have the oppositely charged amino acids which stabilize them. So these are the 5 different type of constraints which affect the stability. Okay. Now we are moving to the last part of today's discussion, and I am going to show you a small table which is about the common and uncommon secondary helical structures which are found in proteins. So you can see here, this is the secondary structure, right-handed alpha helix. It has a phi degree and psi degree of -57 and -47. Then we have a right-handed 310 helix. Okay, so 310 helix we call it, and they have the phi and psi degree of -49 to -26 degrees. Then we have the right-handed pi helix. They are, uncommon these structures. They have -57 and -70. Then we have the left-handed polyglycine 2 and polyl-proline 2 helices, and they have minus 79 to 150 phi and psi. And then we have the left-handed alpha helix, which is 57 and 47. So in this, the right-handed alpha helix, it is the most common, and other are uncommon structures. So that is not the scope of today's discussion. So we will just pause our discussion here, and I hope you have got a clarity about the alpha helix and how they are stabilized within the protein. And in the next class we will discuss about the beta pleated sheets, which are the again a second type of secondary structures. Till then, you take care, and I'll see you in the next class. Bye bye.